Hello students. So we will continue NCERT chapter 10, which is Biomolecules, and today is theory part 4. And we are going to see NCERT section 10.1.3 to 10.1.4. This is disclaimer. So let us see the objective of today's lecture. So today we will explain the characteristics of biomolecules like carbohydrate, proteins and nucleic acids and hormones. And we will classify carbohydrates, proteins, nucleic acids and vitamins on the basis of their structures. Now according to NCERT, the sections which we will cover today is 10.1.3 disaccharides. We will see 10.1.4 polysaccharides, and at last we will summarize what we have studied in today's lecture. So first we are starting with disaccharides. So first let us see the introduction. So first let's define what is disaccharide. So a disaccharide is a carbohydrate that forms when two monosaccharides, which are simple sugars, they join through a special connection which is called as glycosidic linkage. Okay, so they are joined together by glycosidic linkage, or we can say glycosidic bond. Now if you see the name of the disaccharides, the name itself gives a clue about its structure. So if you see over here di means two, saccharide means sugar. So simply I can say disaccharide means two sugars that are joined together. So it is important to note that disaccharide is a type of sugar. This means it shares properties with other sugars like being sweet and soluble in water. So a common example of disaccharide is lactose. So lactose is the sugar which is found in milk, and it is made up of two monosaccharides. So what are those two monosaccharides? They are glucose and galactose. So if you'll see over here at this diagram, so this shows that how a disaccharide, which is a lactose, it can be broken down or hydrolyzed using enzymes. So hydrolysis, what do I mean when I say hydrolysis? So it is a process where water and enzymes help split the disaccharide back into two original monosaccharides. So if you'll see hydrolysis of disaccharide, which is lactose, with enzymes lactase, it splits into two monosaccharides, which is glucose and galactose. Okay. So to sum up, I can say that disaccharides, they are treated with dilute acids or enzymes. They break apart and yield two molecules of monosaccharides. So this process is essential for our bodies to absorb and use the energy which is stored in these sugars. Now we will talk about glycosidic linkage in disaccharides. Okay. So first we see that glycosidic linkage is a covalent bond that actually the actual linkage happens through an oxygen atom. So this oxygen bridges the two monosaccharides units, connecting them together. Okay, so the connection, or you can say the actual linkage, it happens through this oxygen atom. Now let us see how this bond forms. So the process is called as condensation reaction, which means that a water molecule is removed as the two sugar join. So when two monosaccharides come together, they lose one water molecule, which is H2O, and that creates a glycosidic linkage. Okay, so you need to remember that it forms through condensation reaction. Now this is also known as oxide linkage or ethereal linkage. Okay, you can either call it oxide linkage or ethyl linkage. Now, if you see over here, this is the formation of a disaccharide, which is a sucrose, through glycosidic bond. Okay, so this is alpha D glucose and this is beta D fructose. So it undergoes condensation reaction where we will lose a water molecule. So OH from here or hydrogen from here, this H2O gets lost, and this is the glycosidic linkage. Okay, you can see which is marked with red. This is a glycosidic linkage. So you can see this links two monosaccharides, which are alpha D glucose and beta D fructose, through this oxygen atom, which is marked in red. Okay, so you will see this forms sucrose through the glycosidic linkage. Okay, so this is what it is, a glycosidic linkage. Now we will see the types of disaccharides. So first, two monosaccharides units join to form a disaccharide. Okay, so it is formed by the combination of two monosaccharide unit. Now, important thing is that the monosaccharide unit may be same or it may be different. So disaccharides, there are of two types, depending that if monosaccharides unit are same or monosaccharide units are different. So first we will specifically talk about type 1. In type 1, the monosaccharide units are same. For example, maltose. So both monosaccharides units are alpha D glucose. So if you look over here, these are two alpha D glucose which are joined through the glycosidic linkage. Okay, so this is a structure of a maltose. Okay, so if you'll see two monosaccharides, they are same. They are alpha D glucose. Okay, so both of the monosaccharides are same. So we get maltose, which is a disaccharide. So disaccharide with the same monosaccharide unit. Next, type 2, if monosaccharide units are different. Example, sucrose. Okay, so in sucrose, one monosaccharide unit is alpha D glucose and another one is beta D fructose. So if you will see, this is a sucrose which is joined together by two monosaccharides unit which are different through glycosidic linkage. Okay, so this is alpha D glucose and this is beta D fructose, and they are joined through the glycosidic linkage, and this is a disaccharide which is a sucrose. Okay, so this is disaccharide with same monosaccharide unit and this is a disaccharide with different monosaccharide unit. Alright. Now we will see the examples of disaccharide. So we will list some important disaccharides. First is sucrose. Now sucrose is commonly known as cane sugar. Next is maltose, which is also called as malt sugar. Next is lactose, which is called as milk sugar. So one by one we will discuss about all these examples of disaccharide. First we are starting with sucrose, which is called as cane sugar. So first let us look at the key points of sucrose. Now first of all, let us see how sucrose is formed. So sucrose is produced through a condensation reaction, which means two molecules join together and release a molecule of water. So in this case, an equal amount of alpha D glucose and beta D fructose, they combine to form sucrose. So it is equimolar mixture. That means in equal amount we take alpha D glucose and beta D fructose and we form sucrose. So you will notice that the two monosaccharides unit in sucrose, they are joined together by a special type of bond which is called as glycosidic linkage. Okay, so this is a type of a covalent bond that connects the sugar molecules, allowing them to function as a single disaccharide. Now, if you look more closely towards this glycosidic linkage in sucrose, so this bonds form between carbon 1 of alpha D glucose and carbon 2 of beta D fructose. Okay, so this is important that it forms, this is a linkage between carbon 1 of alpha D glucose and carbon 2 atom of beta D fructose. Okay, so this specific connection is what gives sucrose its unique properties and structures. So the numbering of the carbon atoms, it is very important over here, which is C1 of alpha D glucose and C2 of beta D fructose. So this is alpha D glucose and this is beta D fructose. Okay, so if you'll see, this is the Fischer projection and this is the Haworth projection. So if you look at the Haworth projection, we will see that this is this is carbon 1 of alpha D glucose and this is carbon 2 of beta D fructose, and they are joined together by this glycosidic linkage. Okay, so this which forms a sucrose. Okay, so this is important. You have to remember how is this linkage formed, or which carbons of alpha D glucose and which carbon of beta D fructose they link together to form the sucrose. Okay. Now if we talk about where sucrose is found in nature, then sucrose occur naturally in plants like sugarcane, sugar beets, dates, and even in honey. Okay. Talk about properties of sucrose. First, it is a non-reducing sugar. Okay, so a non-reducing sugar, first of all, what is a non-reducing sugar? So a non-reducing sugar is a carbohydrate that lacks a free aldehyde or ketone group. Okay, and therefore it cannot act as a reducing sugar, and it cannot act as a reducing agent. Okay, so if you notice at this diagram, you will see that the reducing group, which is the aldehydic C1 of glucose and ketonic C2 of fructose, they are involved in the linkage. Okay, so if you will see, this is the carbon 1, which is the aldehydic C1 of glucose. In glucose, we have aldehyde group at carbon 1. So this is the aldehydic C1 of glucose, and in fructose, we have a ketonic group. And this is ketonic C2 of fructose. Okay, and these are joined together by this glycosidic linkage, and this forms the sucrose, which is a non-reducing sugar. So you need to remember, a non-reducing sugar lacks a free aldehyde or ketone group, and therefore they don't act as a reducing agent. So it is important to note that non-reducing sugars do not have an OH group at the anomeric carbon. Okay, so we can say that sucrose, they do not have a free OH group at its anomeric carbon. Okay, so this structural feature is what prevents it from acting as a reducing sugar. So when you test sucrose with reagents like Benedict or Felling solution, you won't see the color change that indicates a reducing sugar. Okay, so you need to remember that they do not have a free OH group at the anomeric carbon. So we can summarize that sucrose is a non-reducing sugar because its structures locks up the reactive group that would normally allow it to reduce to other substances. Okay. Now looking forward towards the properties of sucrose, first we will talk about hydrolysis. So hydrolysis is a process where compound react with water and breaks down into smaller compounds, into smaller molecules. So in the case of sucrose, this can happen when we add a dilute acid or we use an enzyme which is called as invertase. So in hydrolysis with dilute acid or enzyme invertase. Now when sucrose undergo hydrolysis, it splits into two simple sugars, that is D-glucose and D-fructose. Importantly, these are produced in equal amounts. So that is why we call it an equimolar mixture. So if we look at the chemical reaction of sucrose on hydrolysis with dilute acid or enzyme invertase, it gives D-glucose and D-fructose and gives equimolar mixture of both of these, D-glucose and D-fructose. So now let us talk about the next property, which is invert sugar. Okay, so the mixture which is formed after sucrose hydrolysis is called invert sugar. So you will see that first sucrose itself is dextrorotatory substance with a specific rotation of 66.5 degree. So dextrorotatory means it rotates the plane polarized light to right. Okay, so if you will see, the specific rotation over here of D-glucose is 52.5 degree, and for fructose, L-fructose, it is 92 minus 92.4 degree. Okay, so if you will see, when we mix these two products, the overall effect is that the solution becomes levorotatory. That is, when this resulting mixture, this which is D-glucose and L-fructose, so when we mix this product, the solution or resulting mixture is levorotatory, because the negative rotation from fructose outweighs the positive rotation from glucose. Look at the negative rotation of fructose, it is minus 92.4, and for glucose it is 52.5. Okay, so this negative rotation from this fructose, this over, or you can say outweighs this positive rotation. So this is a great example of, you can see how combining the substances, they can change the physical properties of solution. So you need to remember that the resulting mixture is levorotatory. So this change in the direction of optical rotation is called inversion of cane sugar. So the term inversion refers to the switch from dextrorotatory to levorotatory. Okay, earlier it was dextrorotatory, and now the resulting mixture has caused it to become levorotatory. So this phenomenon is called as inversion of cane sugar. Okay, so the final mixture of glucose and fructose is called as invert sugar. So the product mixture is called as invert sugar. Okay. Now we will talk about maltose, which is also known as malt sugar. So look towards its key points. So first notice that maltose forms when two glucose molecules join together. So the process is called condensation, where a water molecule is removed as two glucose units combine. Okay. So if we focus on the type of bond that connect these glucose molecules, so it is called as glycosidic linkage. So especially the bond forms between carbon 1 of alpha D glucose and carbon 4 of another alpha D glucose. Okay, so glycosidic linkage between C1 of one alpha D glucose and C4 of another alpha D glucose. So if you look over there towards its Fisher and Haworth projection, this is alpha D glucose and this is also alpha D glucose. The resulting structure is maltose, and there you will see the glycosidic linkage between carbon 1 of alpha D glucose and carbon 4 of another alpha D glucose. Okay, so this is the glycosidic linkage, and this is how maltose is formed. So you will see that maltose, it is found naturally in foods like wheat, barley, rice and corn. So these grains are a very common source of maltose in a diet, especially after processes like germination or fermentation. Okay. Now let's see about the properties of maltose. First, maltose is a reducing sugar. Okay. So, the key reason maltose is a reducing sugar is because the hemiacetyl group at carbon 1 of the second glucose unit is free. Okay, so you need to remember that there is a hemiacetyl group at C1 of the second glucose unit, which is free. And what is a hemiacetyl group? It is you will see in hemiacetyl group, you will see that a carbon is bonded one to an OR group and one to an OH group. Okay, so OR group and OH group is bonded to same carbon. So you will see that the anomeric carbon has an OH group. So you will see that at this carbon 1 of second glucose unit, we have an hemiacetyl group. So if you look at this carbon, this has one OH group, which is this OH group, and it has one OR group. Okay, so you will see that there is a free OH group at this anomeric carbon. So this is an anomeric carbon and there is a free OH group at this anomeric carbon. Okay, so you will see that the OH group is attached, and the anomeric carbon, what is an anomeric carbon? The anomeric carbon is the carbon atom that was part of the carbonyl group in an open chain form of glucose. So this OH group is what allows maltose to act as a reducing sugar. Now, because of this structure, maltose shows reducing properties and it also exhibit mutarotation. Mutarotation means that the sugar, they can change its optical rotation as it switches between different forms in the solution. Okay? So, if we talk about what happens when maltose undergoes hydrolysis. So, we will see that when we hydrolyze maltose using dilute acid, one molecule of maltose reacts with water to produce two molecules of glucose. So, this is a very common reaction in digestion where enzymes or acid break down complex sugars into similar ones, into a simpler one that our bodies can use. Okay, so you will see on hydrolysis the given reaction. You will see this is maltose. When it's react with water in the presence of an acid, it gives two molecules of glucose. Okay, so this reaction shows that how maltose is converted into glucose. Now we will talk about lactose, which is commonly known as milk sugar. So first we will discuss about the key properties of lactose. First we will see that lactose forms when it forms through a condensation reaction between two simple sugars, which is glucose and galactose. Okay, so condensation, that the two molecules, they join together and release a water molecule. Now, if we focus on what type of bond that connects these sugars, so this is a glycosidic linkage in lactose. So the glycosidic linkage in lactose is between the carbon 1 of beta D galactose and carbon 4 of beta D glucose. So this is called beta 1,4 linkage. Okay, so the numbers tells us exactly where the carbon atoms are involved in the bond, and the beta over here, it refers to the specific orientation of the sugars. So it is carbon 1 of beta D galactose and carbon 4 of beta D glucose. So if you see, this is the Fisher projection and this is the Haworth projection. Okay, so if you carefully look, then there is a carbon 1 of beta D galactose and we have carbon number 4 of beta D glucose, and they join together through a glycosidic linkage and they give us a lactose. Okay, so lactose, it is found naturally in foods like milk, yogurt and cheese. So these are the main dietary sources for most of the people. Okay, now we will talk about the properties of lactose. First, we see that lactose, it is a reducing sugar. Okay, so the glucose which is the part of the lactose, it has a free hemiacetal group at carbon 1. So this free group is what allows the lactose to act as a reducing sugar because it can open and react with other substances. Okay, and you need to remember it that the presence of a free hemiacetal or aldehyde group is a key for the reducing behavior. Now, if we talk about because of that free hemiacetal group, lactose is classified as a reducing sugars. This means it can participate in reactions like Benedict test, where it changes the color of the solution by reducing copper ions. So if you see over here, we have a free hemiacetal group at carbon 1. So this is the carbon 1 and we have a free OH group at the anomeric carbon. Okay, so this is what makes the lactose or reducing sugar. Now if we talk about what happens when lactose undergoes hydrolysis. So when one molecule of lactose is hydrolyzed, it produces one molecule of glucose and one molecule of galactose. So you will see the chemical reaction. Here we have a lactose. When we hydrolyze the lactose, it gives beta D galactose and beta D glucose. Okay, so this is the hydrolysis reaction of lactose. Now we will see towards the introduction to polysaccharides. So first we will see that polysaccharides, they contain a large number of monosaccharides unit. So monosaccharides units are simple sugars like glucose, and when many of them join together, they form the bigger molecules which is called as polysaccharides. So first let us see the key points. So first notice that the polysaccharides, they contain a large number of monosaccharides unit. Okay, and these monosaccharides unit, when they join together, they form bigger molecules called polysaccharides. So we will see how these units are connected. So the monosaccharide unit in polysaccharides, they are joined together by special type of bonds which is called as glycosidic linkages. Now polysaccharides, they mainly act as a food storage or structural materials in living things. Now we will talk about some important examples of polysaccharides. So first one is starch, next is cellulose, next is glycogen. Okay, so these are some important polysaccharides. Now we will discuss about important polysaccharides. First we will discuss about starch. So starch is a complex carbohydrates and a polysaccharide which is made up of many glucose units. It is one of the most important sources of energy in our diet. So starch is a polymer of alpha D glucose units which are joined together by the glycosidic linkages. That means that many glucose molecules are connected in a specific way to form the starch molecule. Okay. Now the starch, they serve as the main storage polysaccharides in plants. If we talk about for humans, then starch is the most important dietary source for the human beings. Okay, so for most important dietary source, starch is found in many foods including cereals, roots, tubers and some vegetables. So when you eat things like wheat, potatoes or corn, that means you are consuming starch. Continuing to important polysaccharides, we will see the components of the starch. So starch has two main components. First is amylose. So and the second component is amylopectin. Okay, so the components of starch are amylose and amylopectin. It is important because in many exams it is asked about the components of starch. Okay, so you should know it is amylose and amylopectin. First we will see about amylose. Now amylose is water soluble component. Okay, that is 15 to 20%. Amylose is made up of long unbranched chain of glucose units. Specifically, it contains between 200 to 1000 alpha D glucose molecules, and these glucose units, they are connected by the glycosidic linkages between the carbon 1 of one glucose and carbon 4 of the next, so which we call it as C1 to C4 glycosidic linkage. So if you look at this is the structure of amylose. So you will see over here, this is a long unbranched chain in which we have 200 to 1000 alpha D glucose. So you will see this is connected by carbon 1 of one glucose unit and carbon 4 of another glucose unit. So this is one alpha link. So this is a alpha link. Similarly, this is connected by carbon 1 of one glucose and carbon 4 of another glucose. This is also an alpha link. So it is C1 to C4 glycosidic linkage. So this is a structure of an amylose. Moving on forward, we will talk about amylopectin. So amylopectin is a large branched polysaccharide which is made up of glucose unit, and it is the primary component of plant starch, forming its structures with chains linked by alpha 1,4 and alpha 1,6 glycosidic bonds. So you will see that amylopectin makes up about 80 to 85% of starch, and it is water insoluble. So water insoluble component. Now if we look at the structure of the amylopectin, then amylopectin is a branched chain polymer which is made up of alpha D glucose units. Now, if we look at the linkages, the main chains, they are linked by C1 to C4 bonds, which means the first carbon of one glucose connects to the fourth carbon of the next. And branches form through C1 to C6 glycosidic linkages, where the first carbon of one glucose attaches to the sixth carbon of another. Okay, so this is linear chain formed by C1 C4 linkages with branching occur at C1 C6 glycosidic linkages. Okay, so if we look at the structure, this is C1 and C4 linkage, and you will see the branching occurs at C1 and C6, which is the glycosidic linkages. Okay, so this is structure of amylopectin. Remember C1 C4 linkage and C1 C6 glycosidic linkage. Okay, so this is the alpha link and this is the branch at C6. So this is the structure of amylopectin. Now moving on forward, we will talk about cellulose. So cellulose is a complex carbohydrate or polysaccharide that is most abundant organic polymer on earth. So it is an important structural component of cell wall of plants and for some type of algae, bacteria and other organisms. So for cellulose, it occurs exclusively in plants. It is a plant specific polymer. So cellulose is most abundant organic substance in plant kingdom, largely due to its prevalence in plant. Now cellulose is the main component of plant cell wall. So the cell wall gives plant cells their shape and rigidity. So cellulose is crucial for the plant structure. Now we will talk about the composition. So cellulose, this is a straight chain polysaccharide, which means it is a long chain which is made up of repeating sugar units. Specifically, it is made up of beta D glucose, which are joined by beta 1,4 glycosidic linkage. So you will see this is the structure of cellulose over here, which is composed of beta D glucose, and they are joined by beta 1,4 glycosidic linkages. So this is beta links over here. Now moving on to the structure of cellulose. So you will see that the straight chain of cellulose, they lie side by side and they are held together by hydrogen bonds. And these rigid fibers are why humans can't digest cellulose. We lack the necessary enzyme to break the beta 1,4 linkages. So that is why cellulose passes through a digestive system as fiber. Okay, so this is why we cannot digest it. Now, moving on forward, we will talk about glycogen. So, the first is that glycogen is a storage polysaccharide which is found in animal bodies. So, that is when animal stores excess glucose in their bodies, it is stored in the form of glycogen. So you will see this is a storage polysaccharide in animal bodies. So glycogen act as a storage polysaccharides. This means animal uses glycogen to store extra glucose, which can be broken down later when energy is needed. So glycogen, this is also called as animal starch. So if we look at where glycogen is stored, it is mainly stored in liver, muscles and even the brain. So the liver acts as the main storage site, releasing glucose into the blood when needed, while muscles, they use glycogen for quick energy during the activity. Interestingly, glycogen isn't just found in animals, this is also present in fungi and yeast. Okay? Now moving on forward, if we talk about the structure of glycogen, then the glycogen structure is similar to amylopectin. So both are made up of glucose units which linked together with branches. However, glycogen is even more highly branched than amylopectin. Okay, so only it is more branched. Now if we talk about how glycogen, this is used in metabolism. So, when the body needs glucose, special enzymes break down glycogen into glucose molecules. So, this process is called as glycogenolysis. Okay, so this is an important, this is especially important during fasting, exercise, or even between the meals, ensuring that our cells always have the energy they need. Now, at last, we will summarize what we have studied in today's lecture. So, first we discussed about disaccharides. They are carbohydrates that, upon hydrolysis, give two monosaccharides. This means that when they react with water, they break down into simpler sugar. So, first we will see that disaccharides can be broken down or hydrolyzed to give two monosaccharides. And these two monosaccharides can be same or different. Now, how these monosaccharides are joined together? They are connected by a special type of bond which is called glycosidic linkage, and this is also called as oxide linkage. So, some common examples of disaccharides include sucrose, maltose, and lactose. Then we discussed about sucrose, which is called as cane sugar. So, in sucrose, the link is between the first carbon of glucose and the second carbon of fructose. So, glucose C1 to fructose C2. This is called as alpha 1 beta 2 linkage. And important property of sucrose, it is a non-reducing sugar. This means that both of its reducing groups, they are involved in the linkage. So, it does not react with certain chemicals that test for reducing sugars. When sucrose is hydrolyzed, it forms a mixture which is called invert sugar. This is because the direction in which it rotates the plane polarized light changes from right, which is dextro, to left, which is levo. Then we talked about maltose, which is called as malt sugar. So in maltose, the linkage is alpha 1,4 bond. This means first carbon of one glucose is joined to the fourth carbon of another glucose. So maltose is a reducing sugar because it has a free hemiacetal group. Then we talked about lactose, which is a milk sugar. So lactose has beta 1,4 linkage, which means the first carbon of galactose is joined to the fourth carbon of glucose. And maltose is like maltose, lactose is also a reducing sugar because it has a free hemiacetal group. Then we talked about polysaccharides. Okay, so disaccharides are those. Polysaccharides, we talked about that large number of monosaccharides units, they are joined by glycosidic linkages. Okay, so they are made up of large number of monosaccharides. So these are simple sugars like glucose, and they join together by special bond which is called glycosidic linkages. Now, polysaccharides serve two main purposes. They act as food storage material and as structural materials in living things. So, some common examples of polysaccharides include starch, cellulose, and glycogen. And then we talked about starch. In starch, we saw that starch serves as the main storage form of energy in plants. So when plants make extra glucose during photosynthesis, they store it as starch for later use. And chemically, starch is a polymer of glucose. This means it's made by linking many glucose molecules together in a chain. Starch has two main components, amylose and amylopectin. So amylose is mostly a straight chain while amylopectin is branched. Then we discussed about cellulose, which is an important polysaccharide. So cellulose is the main component of plant cell wall. It gives plants their rigidity and strength, allowing them to stand upright and grow tall. Okay. Then we talked about it is made up of straight chain beta D glucose units. So the term beta refers to the specific orientation of glucose molecules. And humans cannot digest cellulose because we lack the necessary enzymes that are required to break those beta linkages. That's why the cellulose passes through a digestive system as fiber, which helps us with digestion but not providing any energy. And then we talked about glycogen. So glycogen is stored mainly in the liver and muscles of animals. When the body needs energy, it breaks down glycogen into glucose, which can then be used by the cells. Okay, so structurally, glycose is similar to amylopectin, but it's even more highly branched. So the branching allows for the rapid release of glucose when energy is needed quickly. Okay, so thank you students for your attention. I hope this lesson was helpful for you. Thank you.